Hello and good morning, Group 2 IB1 Biology. This morning I won't be joining the class on a live Google Meet like we always do, but I'm going to be sharing in real time the activities that I want all of you to carry out today over a period of about 1 hour. And I want you guys to do this synchronously, which means you're going to work together as a class. As stuff is shared on this lesson, you would be putting it on your personal Hangout as it progresses. So it won't be that you can do this later on. You've got to do this now. Even though I am not participating directly in class, I would be able to see your posts as they come on Hangouts, and I might not be able to reply until later in the day, but at least I would be able to see that you are posting, and of course I can always see the time of the posting. And it's really important that we get together as a community of learners right now and do all of this together at this point in time. While it's useful to have asynchronous activities, it's also important that we meet as a community and do things together in the same place at the same time. So today we are scheduled to have that. I can't make it, but you guys can all make it. And I want you to collaborate with each other as needed, but to share each question that I'm going to ask, each activity, on your personal Google Hangout. Our purpose today, of course, is to look at section 2.6, the structure of DNA and RNA, looking specifically at these understandings here, primarily. Of course, the big picture with the nature of science is using models as a representation of the real world. Watson and Crick elucidated, or they figured out and presented, the structure of DNA, largely with the use of models. Models, yes, but they also got a bit of help from the work of a woman called Rosalind Franklin. You might want to look up and read a little bit about the work of Rosalind Franklin as well. Now there's a link that I've put on Hangouts also to watch a video about the structure of DNA itself at a more macroscopic or a big level. And it's useful to watch that before we even get started with this molecular structure of DNA and RNA today. So first up, I want to ask everybody to get your paper and pencil and to draw this structure. Just as you see it here, draw it on paper with pencil, take a photo of it, and post it on Google Hangouts as your number 1 activity for today. Once you complete that, the next activity is to talk about what this is called and why we call it that. Well, as you can see, the structure here is a pentagon. But important to note, just as I pointed out with glucose earlier in this section, that this pentagon has an oxygen as part of the ring, but that oxygen replaces one of the carbons, but a 5th carbon is located outside of the ring. This carbon here is designated as carbon atom number 1. So you've got 1, 2, 3, 4, and 5. Knowing particularly that this is carbon 5 and this is carbon 3 is useful for discussing more details about the structure of DNA and RNA later on. So draw this structure and write out its full molecular formula. This here is its structural formula. What is its molecular formula? How many carbon atoms does it contain? How many oxygen atoms does it contain? And how many hydrogen atoms does it contain? Once you write that out, you should notice that it is C5, H10, O5. This is a pentose sugar or a ribose sugar. and it is one of the building blocks of a bigger unit called a nucleotide. There are two types of pentose sugars that we are going to consider as we look at DNA and RNA. RNA stands for ribonucleic acid. Ribo. Ribose is a synonym for pentose. It's a 5-carbon sugar. DNA, deoxyribose. So that immediately suggests that there's something to do with the removal of an oxygen, and that is a profound difference between DNA and RNA. The 5-carbon sugar that is part of DNA molecules is a little different from the 5-carbon sugar, the ribose, that you find in RNA. What's that difference? That difference is with one oxygen less. So DNA contains a pentose sugar or a ribose sugar that has one oxygen less on carbon atom number 2, and that makes it deoxyribose. So we've got two sugars that we need to consider. I want you to draw this one also on your hangout and write this, its full molecular formula. Take a note, please, of the difference between ribose sugars and deoxyribose sugars. So this pentagonal structure, whether it's DNA deoxyribose or RNA ribose, is part of something a little bit bigger. And that thing that's a little bit bigger is called a nucleotide. Now I would like you to take out another sheet of paper, move to another page in your notebook, and draw at the bottom of the page or at the top of the page, leaving enough room to add more. I want you to draw this diagram, please, and also place a copy of it on your Hangouts right now so that I can know that you are out there participating in this lesson as I speak. So post this on your Hangout. This is a way of representing the structure of a unit called a nucleotide. And why I call it a unit is because it is one of the basic building blocks of a molecule that is much bigger. A molecule that is much bigger and made up of smaller repeating units. You might recall what that is known as well as you might not. But the answer to that question is it's called a polymer. So both DNA and RNA are huge molecules made up of these smaller units called nucleotides attaching to other nucleotides. So the difference, one of the differences between DNA and RNA lies right here in this 5-carbon sugar. But we're going to look at two more differences today. Before we get to that though, I want you to attach another nucleotide to this one, and then attach a third one and a fourth one and the fifth one, going down your page or going up on your page. But how are you going to do that? You are going to do it following this. Where you have carbon atom number 5 here, you will allow another nucleotide with its carbon atom number 3 to plug in here. Now there's a condensation reaction happening here. Like we spoke about earlier in this section, there's a condensation happening here, and a phosphodiester bond is formed with the 5' end of this nucleotide plugging in to the 3' end of another nucleotide, 5' to 3', and then you keep doing this until you get a chain of 5. Each of them 5' to 3', 5' to 3', and then you would be making a chain where DNA or RNA, we haven't defined what they are yet, whether it's DNA or RNA, each nucleotide unit of that nucleic acid joins by the 5' end of one, plugging into the 3' end of another one, like so. So by this point, you should have a number of nucleotides assembled in a straight chain like this here. Now you might notice that I've got nucleotides assembled on this side as well, and they're all linked up by these lines. We are not ready for that at this point. Now I want to talk about what we have inside of this rectangle. What does the circle represent? It's a P. That P actually is phosphoric acid molecule. Phosphoric acid molecule joins to an OH here by a condensation reaction, and it gets attached here. Then another complicated structure here containing nitrogen attaches to the 5-carbon sugar. These are referred to as nitrogenous bases. So a nitrogenous base, a 5-carbon sugar, and a phosphate, these are the components that make up your nucleotide. And what you have drawn so far, you have drawn some nucleotides making up a chain of 5 going from 5' end of one plugging into the 3' end of another. I've made this long chain here, and I put 5' at the top because that's carbon 5, and I put 3' down at the bottom because that's carbon 3. Now I want you to keep your chain of 5, and opposite to that chain of 5, I want you to draw another chain of 5 coming down this way. So like 1, 2, 3, 4, 5 coming down this way here. But as you draw it, I want you to realize what I've done. And if you have problems drawing it, you just turn your paper upside down. By turning it upside down, what you are really saying is that one unit goes this way and the other one comes the opposite way. That is referred to as an anti-parallel structure, with one chain going 5' to 3' and the other one going 3' to 5'. These bases here, nitrogenous bases, they go by a range of names. Actually, 5 names. 4 of them can be found only in DNA. 3 of them are found in both DNA and RNA. But there's a 4th one that's only found in DNA structures. Structures that have deoxyribose sugars. And another one, in place of that one, that's only found in ribonucleic acid or RNA. These nitrogenous bases are known by the names adenine, thymine, guanine, cytosine, and uracil. You might be familiar with these from your studies before. These are the nitrogenous bases. They work like the letters of an alphabet, really, a 4-letter alphabet, to create what we know as the genetic code. But right now, I want you to label on your drawing with your 1, 2, 3, 4, 5. I want you to label this one here as an A. A for adenine. Then I want you to label this one here as a G. G for guanine. This one as a T. T for thymine. This one as another T for thymine. This last one is another A for adenine. These nitrogenous bases have certain specific structures. They arrange themselves in space in a certain way. And for a DNA molecule like this one to form, not only do we need one chain running from 5' to 3' and 3' to 5', but for these bonds here, which are represented by these red lines, they are actually hydrogen bonds. Hydrogen bonds are, of course, weak intermolecular forces, relatively weak when compared to covalent and ionic bonds. We discussed those when we spoke about the water molecule. When you have one DNA strand running, one component of the DNA molecule, I should say, running this way and the other one running this way, the two strands which run anti-parallel to each other are held together by hydrogen bonds. But if this one's adenine, it's only going to have the right configuration to bond to a thymine by two hydrogen bonds. And if this one's guanine, it's only going to be able to bond to a cytosine. So A bonds to T and G bonds to C, or the other way around as needed. So now what you need to do is to draw your full diagram for your 1, 2, 3, 4, 5, as I indicated here, and fill out the corresponding nitrogenous bases on this side, or the complementary base pairs on this side. Notice I only spoke about 4, because this molecule that we're modeling right now is DNA. Once you have one strand of the polynucleotide chain bonding to another strand that is anti-parallel, anti-parallel meaning 5 prime, 3 prime, 3 prime, 5 prime, then that is another sign that you have DNA. So this is the second of two important differences that we are noting between DNA and RNA. First one has to do with the structure of the pentose or the ribose sugar. Second one is that DNA is made up of a double-stranded structure. And the third one has to do with the presence of one of the bases. RNA is always a single strand, but it never contains the base thymine. Thymine T is always replaced by uracil U. But just like thymine can pair with adenine, uracil could match and pair with adenine as needed, and we will see that that pairing does happen when we look at section 2.7 and we look at protein synthesis. So at this point you should have 1, 2, 3, 4, 5. You would have a small piece of DNA. These bases work like a code. Each triplet actually specifies the position of a single amino acid in a polypeptide chain. But that is going to be the subject of section 2.7. For today though, I would like us to look back now at all of these understandings. The nucleic acids, DNA and RNA, are polymers of nucleotides. Please ensure that you know what a polymer is. What is a nucleotide? What are the three components that make up a nucleotide? And how is a nucleotide of DNA different from a nucleotide of RNA? DNA differs from RNA in the number of strands present, the base composition, and the type of pentose. DNA is a double helix made from two anti-parallel strands of nucleotides linked by hydrogen bonding between complementary base pairs. So all of this is what we covered in this morning's discussion. In addition to that, you were required to do some drawings of the simple structure of a nucleotide using circles to represent what? Phosphate. Pentagons to represent either DNA, deoxyribose sugar, or RNA, the ribose sugar. And the rectangle to represent the nitrogenous bases, adenine, thymine, guanine, cytosine, and in the case of RNA, uracil replaces thymine. That thymine is spelled Thymine, thymine. So the last application that we need to consider is the work of Watson and Crick and how they used a model to understand the structure of DNA, exactly how it forms this thing called the double helix. So I would like you to go into your textbook and read a bit about that. But in addition to that, we are going to have a look at this TED Ed lesson where I use not the same model used by Watson and Crick, but a similar one that is of course widely available nowadays. Watson and Crick first proposed this kind of model in 1953, but today students can build models like this on their own, or you can have models like this at your school, and this is what we have here on this video. Now on this video in the TED Ed lesson, I have started off by giving you some information, and I would like you to initially take a screenshot of this and label all of the components as required here. Read your textbook, research on the web about the structure of DNA and protein synthesis, and when you have mastered this topic, unwrap DNA as follows. First of all, do all of this here. And that is our work for today. Once you can do this, you can share that with a screenshot on your Google Hangout. Then you have two things that you can do with your remaining time. Begin working on your document for the lab report, or read ahead to section 2.7 and complete the rest of this TED Ed lesson. I will be joining Hangouts later in the day to see all of the work that you've put on, and I'll be taking attendance by looking at the work that you've put there. If you do have any questions, write them on your personal Hangout and I will get back to you later in the day.